Hi, my name is Fábio Calambianco, and on behalf of the authors of this paper, Natan Andrade and Fábio Faria, I will present this work entitled Improving Similarity Metric of Multimodal MR Brain Image Registration via a Deep Ensemble. We all work by the Federal University of São Paulo, by the Group for Innovation based on Images and Systems. Here is a brief outline of the presentation. I will start introducing the problem, then showing the proposal and goals within it. After that, I will show you the state of the art of the literature. Next, I'll follow with the methodology and the results and discussion of the experiments. And finally, the conclusions and future works. Our work consists of medical image registration. Image registration is a problem where a geometrical transformation moves a source image onto the same space of a target image in order to have their contents overlapping. The procedure requires three main elements: a transformation, which is applied over the source image with some transformation parameters. The parameters and the transformation nature will indicate what degree of freedom we have in changing the source image domain. Then we'll have a similarity metric that computes the potential matching between regions of the transformed and the target image. And finally, we have an optimization process that is a function that evaluates how well the images match according to the similarity metric and tries to improve the registration of the similarity, changing the parameters. The result of the procedure is an output image. It's a transformed image or the source image on the space of the target image. Our work focuses on the similarity metric element of the registration applied to multimodal images. Multimodal images are images that have the same content with different appearance, such as this example here, where we have T1 and T2 images of the brain that are acquired with MRI scanner. We may also have different modalities between CT and MRI images and so on. And in this example, this is the source and this is the target image, and we want to match these three landmarks with different colors here, and those are the similarity maps that are outputted by the similarity metric for each of those landmarks. For instance, for the red cross here that we have, or the red X, we have the similarity map that is somehow close to the best position that is expected. Red images have the higher probability of matching the target image. The other ones are not very good results. And we show here that this problem is very difficult. In this slide, I show you the classical similarity metrics for single and multimodal images. And we are interested in the multimodal similarity metrics. So from the classical literature, we have mutual information and correlation ratio. More recently, deep similarity metrics started to appear in the image registration scenario. This is one of the first proposed methods used for similarity metric calculation based on deep neural networks. The results are not very effective, as we can see here in this column, that it compares with classical correlation methods. and this is the result of the paper. And it still has a lot of false positive results. And here we show two more methods used to compute similarity metrics. They are present together because they are based on CNN architectures. Both were validated in the context of MRI brain images too. Another very interesting work was proposed by Wang that used a CNN autoencoder. As we can see here, this is the target image, the source and the transformed, and this is the result of their work compared to classical features extracted from the image. It's a little bit better, but still didn't achieve a very good result. Finally, here on the left, we see a CapsNet deep architecture, and on the right, we can see a Spatial Transformer Network, STN. They have been used for image registration, but not to compare similarity metrics. And since their nature resembles spatial transformations, we decided to use both of them in our tests and methodology. And those were the existing datasets at the time that we tested for similarity metrics that contained two modalities of the same patient. We used the largest one to train and test our network. That's IXY. On the right, we can see two sample slides of images from this dataset. And of course, images from the same subject appear in either the training or the testing set, but not in both of them. Before running the experiments, we prepared the images. We selected some slides, normalized them both in intensity and pixel dimensions, and then we stripped the brain and eroded the mask. And this procedure, this erosion, is important to use selected patches. We want to select some patches that were completely contained inside the brain. In the right here, we can see some random points that were chosen in the center of the patches. And we decided to use square patches with 13 by 13, 17 by 17, and 21 by 21 pixels. This slide shows our main methodological contribution. We proposed a deep ensemble to compute similarity maps. The slide shows the training process, but after the training, the same steps are taken for validating and testing, except for the input that receives in the testing a single patch from the source image and the entire target image, and it doesn't use the ground truth, neither to validate nor to update the network weights. But starting here from the left, we have, during the training, pairs of patches that are inputs to the network. They may be both from corresponding or non-corresponding regions. Then we use any number of metrics, both classic and deep learning based, to compute several different similarity metrics, resulting in those maps. After that, we may select any subset of those maps and use them as input to our deep ensemble. The ensemble here is also a CNN and an autoencoder. The ensemble has been trained to learn from the maps as their inputs, and our final result as output is also a similarity map. This approach was designed to be feasible both in terms of required space and computational effort. These are the three deep metrics used in our ensemble. We used DNN from paper from Shang. We also used 2D CNNs and CapsNet. Note that they are not very big networks, since we had to test several configurations and also make them part of the ensemble. The second contribution of our paper is proposing a novel evaluation metric. Traditional metrics did not reflect the desirable features of our similarity metric. As the output patch is small with respect to the image, traditional metrics failed to elucidate simple information, such as, did the method point the highest probability at the point of interest? How many false negative regions are there? The idea is to generate two simple measurements. The first one, PPOR, is a binary measurement that indicates if the transformed patches match the target patch and has a similar size to it. That's because some methods assign a high probability to a large area of the image containing the target, but this is not useful. The second measurement is FPOR and is a quantitative measurement that provides the approximate quantity of false positive patches. Again, large patches may count as multiple false positives in this metric, depending on their relative size to the patch size. To achieve the highest score, a similarity metric should output high probability only to the target area that is roughly the same size as the patch. We also used all other traditional metrics, such as the Jacquard and the Euclidean distance, to evaluate the results. Here we show some qualitative results, and our method here, ESB3 and ESB5, got the best results, clearly. Here, MI, we have those two results from the classical literature, and those three are from the deep learning-based ones. ESB3 uses those three maps as the inputs from the first step of the deep ensemble. And ESB5 uses all those 5 inputs. As we can see, ESB3 has only a single high probability location, and it achieves the best result in this case. And here we show our quantitative results. And as we can see, according to both classical metrics, our ensemble got the best results. Also in terms of false positive in our new evaluation metric. CNN21 got a very good result too, and even better than ours in terms of true positives, but it got 7 times more false positive results. So in conclusion, we can see that our proposed ensemble got a huge potential for multimodal image registration. We would like to test our similarity metrics in this context in future works. Also, we may improve patch selection from random to specific locations or more specific locations. And we also want to use other deep neural networks within our deep ensemble, such as GANs, UNets, and VGGs. Finally, we want to try our methodology with other image modalities and other border regions. And also, we want to improve the parameter selection for our evaluation metrics. Those are the future works. And these are the references that we used in our presentation. And finally, we want to thank CAPS for financial support for Nathan Andrade Master Scholarship, and also NVIDIA for donating a Titan V GPU for our research lab. Thank you very much.